Hello, everybody, and thank you very much for coming. I hope you had a great lunch. I'm Irina Balaur, and I'm a researcher at European Institute for Systems Biology in Lyon, France. And today, on behalf of authors whose names are already here, I will try to introduce the way we use Neo4j approaches for data management in systems biology and medicine. So first of all, the European Institute for Systems Biology and Medicine in France aims to speed up the transfer of results obtained through systems biology approaches to industry and to routine patients by studying life complexity through transdisciplinary approaches. And at the EISBM, the team is composed of biologists, computational biologists, physicians, mathematicians, and engineers, and we work together on different long-term joint projects. Also at EISBM, we work on systems before medicine, which means participatory, predictive, personalized, and preventive. And we have partners from both academia and industry domains. And we work together on a European project in order to boost global public health. The website of EISBM is this one, so www.eisbm.org. Some of the recent projects at EISBM include CASIM project, CASIM Europe, which is coordinating action systems medicine, CISCLAD or MEDAL or UBioPred, which is unbiased biomarkers in prediction of respiratory diseases outcomes, and ETRIX, the European Translation Information and Knowledge Management System Services, the project I am currently working on. First of all, about translational medicine. Recently, there was a perspective paper in the New England Journal of Medicine about an initiative of $250 million spent for involving a voluntary cohort of 1 million people and collecting multi-omics data. In UK, the Genomics England has started already a project which refers to collect multi-omics data from and to sequence 1,100 genomes with a focus on cancer and rare diseases. These initiatives basically are motivated by the need, by the requirements of developing new taxonomy of diseases. And basically different diseases are seen as a collection of single conditions, while actually they represent a group of diseases, of subtype diseases. And the new taxonomy proposes looking to disease from molecular features and not based on the clinical symptoms. A new taxonomy is available, it's possible, due to the cheaper collection of multi-omics data. And basically, this new taxonomy will move medicine from a reactive to proactive perspective, and the focus will be to identify the transition between wellness and disease. While it's very important to have translational medicine informatics, there are also some challenges we have to be aware of. So first of all, is integration of multi-omics data or multi-types, which could help for disease subtypes discovery. So basically, there are hundreds of subtypes of asthma, for example. Then it's moving from understanding molecular patterns basically to the disease mechanisms. So we want to explore subtypes and to guide therapeutic intervention and treatment pathways, which will basically aim to give a more individualized healthcare pathways. And finally, a challenge for translational medicine informatics is to develop a platform to be able to facilitate storage, mining, analysis, visualization, exploration of a large patient data set. So what we can say now these days is kind of like the sequencing of a genome is quite affordable. However, the data analysis and interpretation is very expensive. So in order to develop a platform for translational medicine informatics, we need to have a data warehouse where to store and to query clinical and molecular data. And then we have to provide a framework or ecosystem for data analytics, where in translational medicine is defined basically to come closer to the clinicians. So we are computational biologists, or we are biologists, but we want to talk to clinicians. And then data analytics, which could give us more insights on the disease subtypes discovery. And a major component of translational medicine informatics consists of developing a knowledge base, which could give more information on the disease context, and also to make association, like hidden association between different concepts. I was mentioning ETRIX, and ETRIX is a large collaborative European project where so there is a collaboration between research, academia, and also pharma companies. So we can see AstraZeneca, Pfizer, Sanofi, Bayer, Roche, and others. While on the other side is Imperial, CNRS in France, with us, EISBM, CDISC, and others. So I was saying we have so we are in the big data ages, let's say, or era. However, the utility of biodata sets is practically given by the appropriate methods to investigate this data or to explore, basically to give a value to the data. And main components of the ETRIX consist of having a repository for curated data sets, and also an ecosystem for data analytics, which focuses on disease and target identification. Another major component is that ETRIX pays attention to ethical and legal issues related to patient involvement. So on the left side, we can see the ETRIX architecture. I was speaking about framework. A framework which is, we are using Transmart and is study-centric. So basically we have clinical and multi-omics data. Then is the ecosystem and is open access for data analytics with different tools. And then these tools communicate with knowledge-based representation and come back and so on, with coming from external resources. So the question was, if we can use here Neo4j or graph database approach to represent information in the biocentric data knowledge database, which would give us information about biological context of the diseases and also facilitates hypothesis generation. Advantages of graph databases have been discussed during these days, but just to mention briefly. So why we use graph databases approaches for biomedical data, we know that biomedical data are highly connected. So if we imagine like proteins having thousands of protein interactions or sequence similarities among other proteins or involvement in different pathways are unpredictable. So it's basically, we are now working with multi-omics data, but who knows in 2 years or even next month what type of data we can collect. And we also need to infer associations between different concepts, so to move a little bit further towards understanding the disease mechanisms. This is a slide that gives an overview of how we arrange and we use, where basically developed the ecosystem, I would say, for systems biology and medicine. So we follow a multi-scale approach, starting with, we are looking to metabolites, proteins, epigenetics, and I'll describe immediately, up to disease context, pathways, and to patient information. We integrate data from major international resources like Reactome, Human Protein Atlas, Drug Bank, and others. And the knowledge representation base is, for this, we are using Neo4j and Cypher queries. when I'm referring to data management, I'm referring not only to data integration, it's also about data exploration, it's also about data visualization. So either we use Neo4j in combination with Java, with R, with NetworkX, and we also have possibility to share networks using Index, which is a platform for network community sharing. And we work with both standards for biology and so biological and clinical standards, EISBM and CDISC for now. So now I'll start with the first component of our framework, let's say. So it's we call it the protein framework, which is a protein-centric framework. So what are represented by circles are different concepts. So we have basically this protein, which is a biomarker for this disease, or this protein belongs to this pathway, or is a drug target, or has a protein-protein interaction with another protein, or has sequence similarities. So this is an example of how we use the protein framework for contextual information. So basically, we wanted to query and to explore which are the drugs associated with proteins that have sequence similarity with asthma biomarkers. And here there are different resources integrated, so repositories. So it's data from a drug bank, for example, and from disease gene and so on. So what is represented with here is the node represents asthma, and this protein from here is a biomarker, reported as a biomarker for asthma. This protein from here has a sequence similarity with another protein, which is a drug target for these drugs, let's say. So someone now can infer, of course, so it's not saying with, so we are saying with a level of certain predictability, let's say. So I don't say that 100% those drugs will target asthma. It's inferring, so it's hypothesis generation. However, the cipher query here is very declarative, so people can understand it. Where is asthma, what type of relationships I'm looking for, and what concepts are basically involved. Further, we use the protein framework for link discovery, and I'll read here out loud from March 2013. So many of the most recent dual studies have identified loci associated with asthma containing genes that have either no known function or no known function that is easily correlated with disease phenotype. So we spoke with our colleagues, with clinicians that are experts in respiratory diseases, and some of them were interested to see the relationships, the associations between clock genes, which control the circadian rhythm, and asthma. So again, if we are looking here is asthma with biomarkers or highly well-replicated proteins, and these have a protein-protein association with clock gene neighbors, let's say in this way. So now if we can do more, if we want, we can infer more information. So only looking like to protein-protein interaction and to asthma-related proteins. And another case, so I was saying that graph database accommodates well new types of data. So basically we have developed already the protein framework when we saw the Cornish 2013 paper, which contains information on cell-specific interactions. So what we did, we added that information to a graph database. So for example, here I have a generic PPI in protein-protein interaction, while here, for example, I had more information taken from this paper about what is the protein-protein interaction in a specific cell, in alveolar epithelial, or in macrophage, or in varves. Now, just technical details. So I'm using Java to develop, to import and to integrate data in Neo4j framework. And just quickly about, like, we have 20,000 nodes of proteins, and relationships, sequence similarity proteins are like 92,000 and so on. It works really well from what we need. I'll go to the second component, and as I was saying, so now we had already a protein-centric framework. Now we move to the metabolic framework, And we integrated information from ReconX, which is the most comprehensive human metabolism reconstruction published by TL in 2013 in Nature Biotechnology. And basically, we took this information, so it's the human metabolism reconstruction, which is available in flat files stored in EISBM standard format. It's in an XML format file. However, it's difficult to explore if someone was interested, for example, to identify subnetworks around a specific metabolite. Like here, some of our colleagues were interested about arachidonic acid, which is again involved in respiratory diseases, and we model in this way. So arachidonic is a metabolite in a consumption of a metabolic reaction, and then is a production, and so on. This is a small example, like limited to 40 nodes. Of course, we have 9,700 species, which means proteins and metabolites, more than 7,000 metabolic reactions, and so on. So basically, we had information in EISBML. We imported in Neo4j, and some of our colleagues were very happy. And because they were happy, they wanted to share among the community and to use it, and to share with colleagues, to share at conferences, and so on. And for example, Cell Designer is a well-established framework in the biology community. So what I did, I developed a parser, a Java-based parser, to take the JSON output from Neo4j to translate it back to EISBML, and from EISBML it's easily to be imported in Cell Designer and displayed in this way. So basically, the subnetwork you saw it previously is now represented in this way, and some of our colleagues, clinicians and biologists, were happier, I would say. And about sharing, so we are using Index from here, while for Index accepts, for example, C format, so it's another parser, or it's another component of the same parser, which translates to C format and then to Index and to Cytoscape, which again is well established in systems biology. I was saying at the beginning that we also include data on epigenetics. And first of all, so I'll move to the third component of our work. And first of all, I imagine that many of you already know what is epigenetics, but just to briefly mention that epigenetics can be defined as alteration in chromatin structure that affect the gene expression, but not the DNA sequence. So it's basically DNA methylation, histone-modification, and encoding RNAs. We knew about StatEpigen database, which looks the conditional probabilities between genetic and epigenetic interdependencies in the Bayesian network format. So it's basically saying that APC hypermethylated given MLH1 methylated is a probability of 0.89, for example, or I could look here, in carcinoma stage of a colon cancer. So this is, it's not causality, it's conditionality. So we wanted to model and to see what we can do more, kind of like to infer in future computational modeling. So yes, this is a small subnetwork showing, for example, as I was saying, like MLH1 hypermethylated is given this one, there is the probability to have a KRAS mutation. The type of age gives information on the oncogenesis stage, while the probability value is an attribute of the age. And the epigenetic framework can be used to identify highly connected molecular events. So, for example, we know that MLH1, KRAS, APC are key genes in cancer development, in colon cancer development, but what about other genes? And we also can do exploration of a most plausible pathway connected to two major events. So for example, if we look to MLH1 hypermethylation and MCC hypermethylation, is either in this way or can be also in this way. As I am saying, this is conditionality, so it's Bayesian. A Bayesian formula can be switched. It's not causality. We want to extend this framework with data on causality if such data becomes available. So such a representation can inform further probability computation modeling, so network-based, Bayesian network-based, let's say, approaches. And now I'll move to the fourth component of our work, which basically aims to apply a graph database approach to represent clinical data in clinical standard format. And in order to facilitate visualization, but also exploration of clinical and biomarker data. And we are looking for therapeutic area of asthma. And further, such a component will be used to generate hypotheses for development of analysis data model, so ADA model, who is familiar with CDISC approaches. There is big challenges, let's say, here, because clinical data are represented by many or large sets of parameters. And this makes the graph model to be not trivial, let's say, in this way. However, we use an integrative approach, so we started with several domains, and we plan to include more domains like demographic, laboratory, medical history, and so on. Part of this presentation is given basically these days in CDISC European interchange by our colleagues Irina and Paul in Vienna. And this is just a small visualization of a laboratory test, let's say in this way. So you can see here the node labels are CDISC terms. So for example, AECAT refers to adverse event category, but is the CDISC term, and we prefer to keep it in this way in order to be in compliance with CDISC representation. While for relationships, we had to define. So for example, this is a patient which participates in a study, and then this patient has some demographic data, or this patient has a screening, where in the screening, they take some samples, they analyze it, they return with results for those analyses, with the data, and so on. So yes, for patients, for example, we keep information about aging and other characteristics which goes for the patient. So yes, in conclusion, today I was presenting a multi-scale approach for using graph database in systems biology and medicine. And I started with translational medicine, and I will go also with phrase from Bebeck in 2012, which explains that the goal of translational medicine with respect to diverse omics data is to allow a scientist, a clinician, with limited computational skills to connect multiple layers of patient-specific information to arrive to a more informed prognosis. So I was saying from epigenetics, from metabolites, up to the patient level. And we use the Neo4j basic integration, and we use Cypher, which we found expressive, as I said, extensible, and I was speaking about adding new data types. Visual has also nice visual data exploration. However, developing the graph database is not trivial. And basically it's not trivial also because of connecting different layers of information among systems biology, let's say. And another challenge is to make these tools more accessible for clinicians and for biomedical scientists. I'll take couple of seconds to introduce a special interest group meeting in Translational Medicine Informatics, which will be held in Orlando during the EISBM 2016. And if you are interested, you can access the URLs, or you can also write to us an email. We are looking for your applications, let's say. I would like to thank my colleagues from EISBM, our colleagues from Centre de Calcul in Lyon, Ron and Chris for their help, and also to acknowledge access to Neo4j and to databases that we use to interrogate. So it's DiseaseGene, Drug Bank, Human Protein Atlas, ReconX, StatGen, UniProt-ID, and others. And finally, I have to acknowledge, and I would like to acknowledge financial support to ETRIX, our project, and thank you all for your attention. And if you have questions, I'd be happy to answer.